Good afternoon. I'm Linda Cabe Halpern, Dean of University Studies here at James Madison University. I'm pleased and honored to welcome all of you to this very exciting event. Here today are some JMU students, faculty, administrators, members of our Board of Visitors, members of the local community, and some guests from farther afield. We are really happy that you are all with us today. Today's lecture is one of a number of events organized around the opening of this beautiful new Performing Arts Center, and also one of a number of events on the JMU campus throughout this academic year focused on the human genome, a series of events we call the Dance of Art and Science. These events started last August when incoming freshmen read a series of Pulitzer Prize-winning essays on the DNA age by Amy Harmon, and then, led by members of the Liz Lerman Dance Exchange, enacted the genome in a dance covering the entire quad. We have had visiting scholars, art exhibits, poetry, projects within various courses, multiple visits by members of the Liz Lerman Dance Exchange, and this weekend's performances in our main stage theater of Ferocious Beauty, Genome. I have been informed that there are a few tickets remaining for tonight's performance, and if there's anyone here today who would like to claim one free of charge, go to the box office in the grand lobby when this lecture's over. We are extremely honored to welcome today to our campus Dr. Francis Collins, Director of the National Institutes of Health, who will surely provide one of the high points of this year-long series of events. Dr. Collins is not only Director of the NIH, he is also past Director of the National Human Genome Research Institute, where he led the effort to complete the Human Genome Project, the successful mapping of the human genome. His groundbreaking genetic research has led to the identification of the genetic variants associated with type 2 diabetes, the genes responsible for cystic fibrosis and Huntington's disease, and many others. He is a recipient of both the Presidential Medal of Freedom and the National Medal of Science. Dr. Collins is known throughout the world for his groundbreaking work mapping the human genome and identifying the genetic foundations of disease. He is also the author of 2 books addressed to the general public: The Language of God, A Scientist Presents Evidence for Belief in 2006, and The Language of Life, DNA and the Revolution in Personalized Medicine in 2010. What is perhaps less well known is the extent to which Dr. Collins is uniquely able to speak to us of both science and the arts. The best way to introduce this other side of Dr. Collins is to tell you all that the neck of his guitar is decorated with an inlaid design representing the double helix. Many of you know that Dr. Collins is a native of the Shenandoah Valley and grew up in Stanton and Augusta County. And Dr. Collins grew up in an extraordinary household filled with music, theater, dance, poetry, and celebrations of the arts, from the extremely scholarly to the avant-garde to the crowd-pleasing. His father was Dr. Fletcher Collins, Yale-trained medieval musicologist, cultural laureate of Virginia, head of the theater program at Mary Baldwin College, and lifelong advocate for the contemporary performance of medieval music and drama. Dr. Collins' mother, Margaret Collins, playwright and teacher, was quite simply a force of nature. Margaret and Fletcher Collins, for many decades, gathered around themselves an amazing community of artists, writers, musicians, as well as generations of people who appreciated and participated in those events. It is a community I was fortunate enough to grow up in, and so the first time I ever saw Dr. Collins was probably sometime in the late 1960s, and I am sure that he had a guitar in his hand. Margaret and Fletcher Collins founded both the Oak Grove Theater in Augusta County and an organization called Theater Wagon to support the arts both locally and in many other places. We extend a special welcome today to members of the Collins family and members of the Theater Wagon Board of Directors who are with us today. So Dr. Collins, as you now know, has deep knowledge and respect for the performing arts that complements his extraordinary achievements in science and medicine. We are extremely honored to welcome Dr. Francis Collins to the JMU campus, to our beautiful new Forbes Center for the Performing Arts. The title of his presentation this afternoon is Art Imitates Life, the dance of DNA, decoding, and doctoring. Please join me in welcoming Dr. Collins.
Well, what a joy it is to be introduced by Dean Linda Cabe Halpern, a lifelong friend, and whose family is also here, as well as many other friends from the Shenandoah Valley that I have loved over many years, and it's always great to come back to the Valley and to be in this beautiful space. My goodness, what a gorgeous building and a gorgeous hall this is, to be able to talk about science and to talk about art. So really, it's a great pleasure to have been invited to be part of this year-long celebration of the ways in which science and art can work together to enrich humanity. And I think that's really what JMU is trying to do, and succeeding in ways that I really haven't seen in other campuses. So this very inventive, organic, evolutionary process that you've been engaged in this entire year is really wonderful to take part in. So I'm thrilled to have a chance to tell you a little bit from my perspective about where the science of genomics is going, and to engage afterwards in a conversation with some students, and then to be able to see the wonderful Liz Lerman presentation this evening, the Ferocious Beauty genome, which I've had the chance to see a couple times before, but it always evolves. And again, you're fortunate to have Liz and her dance troupe here as part of this, because they are, in my view, one of the most creative groups around that is mixing together the artistry of dance with messages, even messages about science, which many people would have wondered, could that work? Well, if you've seen this, you'll know it works beautifully, and I'm happy to be able to see it again this evening. So what to do here with my time with you? I thought, well, I'd better talk about the science of DNA and where it's going, and how it has touched increasing numbers of people in direct ways in terms of its impact for medicine. But of course, it goes even beyond that in terms of our view of ourselves and what we think we're made of and how we're related to each other. So I won't be able to cover all of that territory in the next little while, but I'm going to take a crack at some parts of it. And I hope at the end we'll have time for some questions, and if you have things you'd like to ask about, please be saving those up. So what is DNA? Well, DNA, of course, is about inheritance. DNA is about the fact that parents pass on properties to their children. Sometimes those are obvious, sometimes not so obvious. I kind of like this opening slide as a way to get us into the mindset of transmission of characteristics from mothers and fathers to their children. Get it? That's all you need to know. Well, okay, so there's probably a little more going on here than just the clothing worn, because that child has also received half of his DNA from mom and half from dad, and with that, a whole host of interesting variants that make us slightly different from each other, even though we are also very much the same. Your DNA and mine, if we laid it out and counted up all the letters one by one that were different, we discover they're relatively few, less than about 0.5% of difference between you and me. So 99.6% of my DNA and yours are identical. But of course, because the genome is a big place, that still leaves room for some very interesting things that make us all unique. Unless, of course, you have an identical twin, in which case you're one of those amazing people who has somebody else on this planet who has the same DNA sequence that you do. So let's talk about the genome and what it is. And of course, you probably have been exposed to various renditions of what DNA is all about in magazines like this, and sometimes they get it right and sometimes they don't. I've noticed, though, that almost every time a magazine like Time or Newsweek has an article about DNA, they always put naked people on the cover. And I wondered about that until I realized there's a simple marketing principle at work here. They know what sells magazines, and it's not DNA. So it was, in fact, a discovery which goes back in terms of about 1944, when we realized that DNA was the hereditary material. But it was not until 1953 that James Watson and Francis Crick figured out its structure, that amazing double helix, which is now the icon that you see everywhere, including in freshman orientation at JMU on the front lawn, because that is, in many of our minds, the way we now think about inheritance is that double helix. But you could also think about DNA as an instruction book, and it's not a bad metaphor. It is an instruction book that's written in a very strange language, mind you. Of course, people think English is strange as well, so maybe we should admire this as a really much more straightforward language, but it's one we're only really beginning to understand how to read. It has only 4 letters in its alphabet. They're chemical bases. We abbreviate them A, C, G, and T, and I can't tell you how many times members of Congress have asked me, why didn't you just call them A, B, C, and D? Well, darn it, it's because they stand for adenine, cytosine, guanine, and thymine, and we didn't want to do violence to the chemical basis of inheritance, but that would be a bit of a sophisticated response for a congressional hearing, so I don't usually say that part. So anyway, A, C, G, and T. So, and that instruction book is made up of a lot of those letters, and it exists inside each cell of your body, which is pretty amazing to think about, because you have about 200 trillion of those cells, and each one of them has to carry the instruction book for life, and that instruction book is about 3 billion of those letters. And that means if you printed it out as a hard copy, which people generally don't do, you would stack the pages one on top of another, and they would be about as high as the Washington Monument. And you have all that information inside each cell of your body, and every time the cell divides, it's got to copy the whole thing. It's amazing this works, isn't it? But it does. And half of that gets passed by the parent to each child, the other half coming from the other parent. Well, the Human Genome Project got underway in 1990, 21 years ago, at a time when many people thought we would probably never be able to read out those 3 billion letters in anybody's lifetime, but a few visionary people said, you know, maybe we could try. And the Genome Project was actually not so widely embraced at that point by the scientific community. A lot of people thought this was foolhardy, that it would never actually be possible to do this in the 15-year timetable that was promised. But a lot of hard work by a lot of very dedicated people kicked in to this effort. Some of the best and brightest of this generation of scientists, and that included engineers and physicists and chemists and biologists and physicians and, goodness, ethicists and theologians, all got involved in trying to see whether we could get this to happen. It was my role to come and lead that effort starting in 1993 and to try to pull together the groups in 6 different countries, some 20 different laboratories, working on this furiously to try to get the job done. And there were times in the early few years where it seemed like this was just never going to happen because our abilities to read out the DNA sequence at the level that we had to was woefully inadequate. But technology came along, and because of a lot of smart people made it come along. And in April 2003, we were able to say, it's done. We have the complete DNA sequence of a reference genome. And people often ask, well, whose genome was it anyway that you sequenced? At this point, this was essentially a patchwork of about 5 or 6 individuals who donated DNA anonymously, so we don't know who they are, and that was intentional. And we did what you would do in that situation, where you tried to see which of those DNA samples gave you the material that you could work with most easily, and out of that came this reference genome, which is great for looking at the 99.5% where everybody's the same anyway. But of course, we wanted to know about the differences. We'll come to those. I have to stop here and give credit, though, to the amazing team that did this, and this is just one photograph of some of the leaders of this effort that basically decided that this was so important and so historic, they weren't going to care too much about who got the credit. They're going to simply make sure this got done. And importantly, also made a decision way back in 1996 that they were going to give the data away every day. Not waiting for any publications, not claiming any intellectual property or patent claims on the material, just give it away, because this information was so fundamentally important to understanding how life works for humans and would be so critical for medicine that you didn't want anything to slow down that process. And so that was the mode they followed, and all of that information then began to get used almost immediately because of that decision. And again, these are real heroes, and for me as the person who had the job of overseeing this effort, it meant that Woodrow Wilson's statement about how you get things done was certainly true for me. Having grown up in Stanton, I always like to quote Woodrow Wilson, "I not only use all the brains I have, but all I can borrow." I could borrow a lot of brains to get this done. Well, that resulted then in laying out all the human chromosomes. That's what you see here. That's chromosome 1 up there, and all the way down to the X and the Y. And on that, we could lay out where all the roughly 20,000 genes were that are encoded within the genome that code for proteins. And a lot of people were pretty shocked to find out there were only 20,000 genes, because they thought we are pretty fancy organisms and we should have had more than that. After all, by the time we got the human genome sequenced, other simpler organisms had had their genomes done. Fruit flies turned out to have about 20,000 genes. Roundworms, too, about 20,000. Rice turned out to have about 30,000. So yeah, if your impression of the importance of humanity was based on the gene count, you were in real trouble at this point. But fortunately, I think we could still look at the reality here that none of those species had sequenced their own genomes, and we had. So there. But rest assured, there are things on your dinner plate this evening that have more genes than you do, so keep that in mind. So anyway, we had the information, but of course we wanted to go forward and see, well, how does it work? And we also wanted to do lots of comparisons with all those other genomes, because evolution is a wonderful laboratory to be able to figure out which parts of the genome have been most important, because they're the ones that are going to change least over time. So we went on to do the mouse genome, 2002. And I'm just going to show you some covers of other genomes that got done. The rat, that's a really awful cover, but it was a good genome. The dog, the chimpanzee, very close to us, the macaque, the honeybee, the sea urchin, for heaven's sake, the platypus, my favorite of all of them. Again, when you line up all these genomes and compare them side by side, you can make some very interesting inferences about which parts have been most crucial, because they vary the least. And if you're looking for digital evidence that Darwin was right about a common ancestor, this is phenomenally compelling in that regard. And we've gone on to sequence the genomes of everything you see in this picture. Now something like 45 different vertebrates have had their genome sequenced, and countless invertebrates. And when you get down to bacteria, it's now over 1,000 that have had their complete genomes determined. The data just keeps pouring out in great abundance. And of course, that leads us to our question about, did it actually contribute something to medicine? I'm a physician. The reason I got excited about genetics and eventually the human genome was the hope that this kind of information would ultimately contribute to the prevention and treatment of disease. Hopefully not quite in the fashion that you see here. It doesn't quite look like the high tech, or nor does it look like it's going very well here in this cartoon. But how are we doing on that? So now I want to switch to talking about personalized medicine and how this understanding of DNA is finding its way into the ways in which we prevent and treat disease. So what is personalized medicine anyway? Well, it is basically the opposite of one size fits all. It is to use an individual's genetic profile and every other kinds of information that you can obtain about that person, including their environmental exposures, to guide decisions about prevention, diagnosis, and treatment. If you are trying to help people stay well, wouldn't it be a good thing to include everything you know about that person so that their individual risks could be focused on instead of this one-size-fits-all prescription, which is often what we've been doing because it was the best we had. If somebody gets sick, wouldn't you like to know about their very specific kind of illness in terms of choosing what kind of treatment to offer? That's what personalized medicine is supposed to be about. So this means that when it comes to therapeutics, it should facilitate the selection of the right therapy for that person. And certainly the advances in this field, many of them we counted on coming from the Genome Project. And I'm going to tell you a little bit about some of those. So one of them comes from this thing called Genome-Wide Association Studies, which is a lot of syllables, or GWAS. So what is this? This is the effort to try to identify where are those hereditary factors that play a role in diabetes or heart disease or high blood pressure. We know they're there because those diseases tend to run in families, but they don't run in families in simple ways that you could explain by a single gene that's gone awry. They must be due to multiple genes, each one of which has a sort of subtle effect on risk, and depending on your whole menu of genetic risk factors that you've inherited, you might tip over into getting that illness or not. So because we had the whole genome, it is now possible to scan across it and try to find these variations that might actually be responsible for disease risks for conditions like diabetes or heart disease. The way this works is diagrammed here. So there's some DNA, the famous double helix. And of course, I said we're all very similar, but we have some differences. So each difference here is a variant, a V. And actually, the way this diagram is shown, it looks like there are a lot of them. Well, actually, there's only really about one every 800 letters of the DNA code that would differ between you and me, but just for the sake of this cartoon, we're looking at a lot of them. Now, most of them don't matter. They fall in a part of the genome that isn't doing very much, has no consequences. But somewhere in there will be a few, oops, like that one, which are actually not so good for you, at least not in our current environment, and may be putting you at risk for some disease, a ticking time bomb, if you will. So wouldn't you like to know what those are for yourself? Maybe you would, maybe you wouldn't, actually. We'll come to that. But it is possible as a research project to go in and find what are the ticking time bombs for lots and lots of diseases. And we didn't used to have that power, and now with the genome we do. So let's see how that works. Most of those variants that we're talking about are actually very subtle. They're not big rearrangements of DNA. They're simply something like that, where you have a C or a G. And if you want to amaze your friends, you can talk about Snips, because Snips are all the rage. A SNP is actually a single nucleotide polymorphism. So that means it's a single letter, a nucleotide, A, C, G, and T are all nucleotides, and a polymorphism just means that it's different between different people in the population. So that's a SNP. And there are about 10 or 15 million of these SNPs in the genome that are commonly variant between people in a group such as this. And the question is, well, which of those are actually ticking time bombs and which ones are irrelevant? So how do you do that experiment? Well, you find a whole bunch of people who are willing to participate in research, who are affected with the disease, and a whole bunch of other people who clearly don't have that disease. And you go SNP by SNP by SNP through all of the genes in the genome to try to find something that looks like a difference. And most of the time, you find, like here in gene A, there ain't nothing different about this particular one. Whether it's green or purple, it is not going to make any difference in the risk of disease. But if you look exhaustively through the whole genome, you might find something like this, where the orange spelling in my cartoon here, which is actually maybe a C, is overrepresented in the affected individuals compared to the unaffected, who seem to have more of the version that's blue, which might be, say, a T. And that would tell you something's going on here, that that particular SNP is associated with disease risk, and therefore it may reside within a gene that is contributing to that disease. And now you know something you didn't know before. So that's what the idea is of a genome-wide association study. Have we done this? Boy, have we ever. We couldn't do this until about 2005 because we didn't have a good catalog of SNPs, but now we do. And it allows us to start reading the book. And that means you scan the genomes of large groups of well-phenotyped individuals. That means you really know their clinical situation. You identify these segments of DNA that show an unequal distribution of variants. And this locates these that are associated with common diseases or quantitative traits, like what's your cholesterol, for instance. And they're also very readily tested and quite cheap to do. So you can do this experiment on thousands of people for an affordable cost. So here's what happens. This was the first success. Now you're looking at the human chromosomes. That's chromosome 1, 2, 3, 4, and so on, all the way over to 22, X and Y. These are schematics. And that red dot there was the first success of a genome-wide association study. And what they discovered there was a variant in a gene called complement factor H in a disease called age-related macular degeneration. Macular degeneration is one of the most common causes of blindness in people past the age of 70. My aunt was affected with that condition and really robbed her of vision. She was a school teacher, and this was a great source of frustration. And we really didn't know what were the factors that played into macular degeneration. And then we found this. And it pointed to a gene that nobody would have guessed would be involved in that disease. And that's one of the powerful aspects of this. You don't have to guess the gene involved. You can just look at all of them and find out what the answer is. So you don't have to depend on a hunch. That was the first one for that disease. Now I'll show you what happened in the next 5 years as this began to be applied to lots of diseases. Each one of those dots you see appearing there is another disease that has had a variant mapped that plays a role in risk. And watch as the years click by here, the dots grow and grow and grow. And this has been an enormous outpouring of data from lots and lots of groups around the world who have used this strategy in order to find now about 1,000 of these. And most of them are big surprises. They are genes that we did not know were going to be involved in that particular disease. And they give us insight into the pathways that are causing these illnesses that are the best hope we've got for the next round of developing therapy. So this is a very exciting moment to sort of move into new territory. Most of these are relatively modest in their risk. We'll come to, in a minute, the possibility of using this information to make individual predictions about future illness in people who are currently healthy. And we can do that, but it's not very precise yet, because we still have not scratched the surface of all of the heritability that must play out in terms of individual risks for disease. So we have that. That's resulted in gene tests growing in numbers for both rare and common diseases. Look and see what's happened here. Back in 1993, there were only about 100 diseases for which testing was available. Now it's over 2,000, and that's going to keep growing. So let me walk you through how this information is now being applied in medicine, and I'm going to pick examples that are not science fiction future. I'm going to pick examples that are happening right now. The first of those is to use this kind of information to diagnose and predict risk. And I just said some of the risks that we've discovered are modest enough that they may not be particularly medically relevant, but some of them are. And so, for instance, I'm going to show you an example about colon cancer where this kind of testing can actually be life-saving. So let's look at a family, which is a family that I know, actually. They live in Baltimore. And here is the circumstance that caused this family to seek attention. This is a pedigree, so this is the oldest generation, grandparents, next generation, and the kids down here. And each of the individuals who's outlined in black here has been affected with colon cancer, including the youngest generation, who are only in their 30s and 40s. So that's very early onset colon cancer, and obviously a big concern here about what's going on. The person who actually came to attention is this woman who, watching what was going on in the family, wanted to know, well, what is her risk and what is her children's risk, and should they be doing something other than just hoping that this wouldn't strike? Well, this is a circumstance where we have discovered the genes that are responsible for this high risk of colon cancer in families of this sort. So it's possible to actually go and get the DNA specifically tested and then find out who got the glitch in the DNA. If you're one of those in a family like this, your risk of colon cancer is 60%. But the good news is, if you start colonoscopy, if you're in that category, at age 30, not age 50, but age 30, and you do it every year, not every 5 or 10 years, you will pick up the polyps that are small and easily removed, and you will live a normal life. And if you don't have the opportunity to do that, again, your chances of colon cancer are 60%, and oftentimes, by the time it's detected, it's too late. So this is not one to pass by. So in this family, that was done. Those are the people who, just looking at the pedigree, are at high risk here of having inherited the same problem that has affected their relatives. And after the test was done, it turns out that 2 of them, the original woman who asked the question and her daughter, are in fact carrying that. And they are now in that colonoscopy kind of program and already have had a couple polyps removed. Whereas this person and this person didn't get the mutation and are at no higher risk than anybody else. So they don't have to go through that exceptionally intense kind of screening process. DNA testing doesn't always give you bad news. Sometimes it can give you really good news as well. So that's an example, a very real one. This is not a terribly rare condition. Something like 1 in 1,000 people is in a family of this sort. And we can now offer those families a lot more than we used to. Another area of application, which is growing rapidly, is the ability to be able to choose drug therapy more precisely than we did in the past, because individuals vary in their response to a drug treatment, and a lot of that is on the basis of DNA. You're probably familiar with the fact that when you get sick and you are given the drug which was the right drug for your disease, it doesn't always seem to turn out the way you want. And that's not just because the doctor made the wrong decision. It's because your particular heredity situation may have made you one of those people who had a bad reaction to that drug or didn't respond positively because you couldn't handle it in the way that other people would have. So here's a good example of how this can happen. You take people with the same diagnosis. Pharmacogenomics simply means drugs and the genome. How do you put them together? And if you actually treat all those people, a lot of them will have a good outcome, but a few will have no response, and some may have a toxic effect. And that's going to be true for most drugs. But we can now start to predict that. Perhaps a particular dramatic example, because it's a drug that is given so frequently, is a drug called Plavix, which is used to prevent blood clotting, especially for people who've had a heart attack or a stroke. But what was observed is that about 30% of people who got this drug were not benefited by it. It didn't help them. What was that about? Well, it turns out there's a gene with a very awkward name of CYP2C19, which has a common variant in it. This is a drug which, it turns out, is inactive in the form you take it in the pill. Your liver has to activate it by an enzyme reaction. Well, if you don't have a functioning version of that enzyme, the drug isn't going to help you. You can't activate it. And that turns out to be the case for about 30% of us. So the FDA has now put on the label for that drug a black box which says to the physician, before you prescribe this drug and assume that it's going to help the person you just gave it to, you should probably check their DNA and see whether they're in that 30%. Because if they are, you better pick something else if you think it's going to help them. So that's pharmacogenomics, and there are now dozens of drugs for which this is starting to be applicable, and a few dozen of them probably in the next 4 or 5 years will have this very much as part of the expectation that before you write the prescription, you check the genome to be sure that's the right prescription for that person. The third area is to use this information for new kinds of therapies. And those could be both in terms of developing a drug based on understanding something about the gene that is contributing to disease, or it could be using the gene itself as the treatment, gene therapy. I'm going to show you a rather dramatic example of gene therapy that has been recently developed for a particular form of childhood blindness. Gene therapy's had quite a roller coaster experience over the last 30 years. Initially, people thought this would be a very direct way to deal with genetic diseases. You just replace the gene that wasn't working with one that did. It's turned out to be much more difficult than that because it's so hard to deliver the gene in a form where it actually ends up getting used and remains stable. But it is starting to work in some interesting and important conditions, and I'm going to show you just one. So let me tell you about Corey. That's Corey with his mom and dad. Corey has a form of blindness called Leber's congenital amaurosis caused by a mutation in this gene called RPE65. None of that matters too much except to say it is a genetic defect. His gene for RPE65 doesn't work. And he was born with normal vision, but as he grew, his vision got poorer and poorer. And now, at about the age of 7 or 8, he is essentially blind. But people have identified the specific problem in this gene and been able, therefore, to create a gene therapy vector that allows the possibility of giving that normal gene back in the eye. Now, what they've done at this point is to treat one eye, and what I'm going to show you is how he can now perform a few weeks after the gene therapy treatment, depending on which eye he's using. First, we're going to look here. This is the untreated eye. So in this case, this is the vision that he's had without any help. And then we're going to look over here and watch him try to navigate this maze with the eye that's been treated. On the floor, you can see these various arrows, and their instruction to him is he's supposed to follow the arrows and not trip over anything, and then ultimately go out the door at the end of this. So let's see how he does, and let's start with the untreated eye.
You don't see any lines on the floor that tell you which direction?
No. You want a clue? He didn't even see anything.
So see, he's unable to even see the lines on the floor because this disease has robbed him of his eyesight. But now, let's see what happens in the eye that's been treated.
Okay, perfect. All right. And you can start whenever you're ready.
That's pretty amazing, and that has been sustained now for 1.5 years, just with that one treatment, and they are considering now treating his other eye. So this is a young guy who was otherwise really headed for a very different future. Let me tell you about a way in which gene discoveries can also be used to develop drugs that are more effective than the standard ones. So I'm going to tell you about Kate. Kate is a cancer patient. Diagnosed with lung cancer, though she's a non-smoker, and actually cancer that was already very advanced by the time it was discovered. The chemotherapy that was offered to her slowed things down a little bit, but not for long. And ultimately, she was facing what appeared to be a hopeless situation until a new drug therapy came along. So I'll get Kate to tell you her story.
When tumor showed up in her liver, doctors didn't give her much hope.
The message I got is make your plans, get your life in order, and enjoy the next few months. And I was enraged.
She said she was enraged because she couldn't imagine how this could happen and there wasn't something else to be done. Well, what happened? She, again, diagnosed at age 44. And despite surgery, radiation, and chemotherapy, the cancer spread. But then she participated in a clinical trial for a new drug called Iressa, which is based upon having understood in certain cancers how it is that mutations in DNA cause cells to grow when they're not supposed to. Because cancer is a disease of DNA. Cancer comes about because of mutations that activate cell growth when it isn't supposed to happen. In this case, Iressa was designed to block one of those activated cancer genes, and it happened to be one that was driving her particular tumor in a very significant way. And as a result, the drug was given to her to see if it would work. And you can see in these images, this is when she started therapy. This is a liver. Those spots that you see there are big collections of cancer cells. 6 months later, those are fading away. Today, well, this is actually now about 8 months out of date, but I've talked to Kate recently. She's doing great. And look at this. This is 8 years. This is somebody who wasn't supposed to live more than a few weeks. And again, this is what can happen when you have the right match between the mutation in the tumor and the drug. Now, why doesn't Iressa work in all cases? Because not all lung cancers have exactly the same kind of DNA glitch that she does. But it is now possible for people diagnosed with lung cancer to quickly check and see whether they're in that subset of individuals, and it's about 15% that would respond to this drug and make sure they get it if that's the case. So those are a couple of examples of the way in which personalized medicine can really provide dramatic responses in terms of therapy. But we're still on the road to this. Now we're moving even more rapidly to use the science of genomics in the practice of medicine. This graph that you see here shows you what's happened to the cost of sequencing, that is reading out the letters, of a megabase, which would be 1 million letters of the DNA code. And back in 2005, that cost close to $10,000 to do 1 million base pairs. Now it's well less than $1, and it's plummeting every day. This is Moore's law, the famous law for computers, which everybody thinks is like the ultimate in technological advances, where things go rapidly down in an exponential fashion. DNA sequencing is outstripping Moore's law rather dramatically. In fact, to the point where we're now having trouble being able to store all the data. Because biology used to be thought of as a not very challenging problem for the computational scientists. Now we've sort of become respectable. We can make terabytes and even petabytes without hardly even trying. And of course, analyzing all that is turning out to be quite a challenge. So this is the way in which the sequencing cost is coming down. And that now puts us in a position to be able, with individuals, to determine their entire genome. Remember, it took us 15 years to get that first one, and it, by the way, cost about $400 million. The cost right now of sequencing a complete human genome is about $10,000, and it will be less than $1,000 in the next 4 to 5 years. So do not be surprised if your own human genome sequence becomes an important part of your medical record in the next half decade, where it can be immediately used by your care providers in making decisions about how to manage your preventive care, as well as decisions such as, is that the right drug for this person, or is that the right dose, all of the pharmacogenomics stuff I talked about. And sequencing has already gotten to the point where applying it across the entire genome has provided some really interesting answers to mysteries. So I'm going to tell you about Nicholas. So Nick here with the spiky hair is now 6 years old, but he developed right after his first birthday something called inflammatory bowel disease, also called Crohn's disease. Very unusual to see in such a young child. Developed really severe consequences of that, with multiple fistulas, the need for many, many surgeries, little solid food, and no idea what was causing this. So the group that was managing his care in Wisconsin decided, well, let's just sequence his genome and see if we can find an answer, because they didn't know what else to do. And they found a mutation in a gene called XIAP, which doesn't mean a whole lot. It turns out that mutations in that gene of a different sort had been described in rare patients who had a blood disorder, but they didn't have Crohn's disease. So this was a bit mysterious. But a few patients who had XIAP gene mutations had responded to bone marrow transplantation. Now, they would not have considered bone marrow transplantation for Nick without this information. But in this situation now, with him growing ever weaker because of all of these problems, they decided to try that. He had his bone marrow transplant last summer, and today, I talked to his mom a couple days ago, he is doing great. He is eating normal food. He has not had a fistula in the last 4 months. He's a totally different kid. Without that DNA sequence information, this never would have happened. So this is another example of how we are moving this technology forward in ways that are pretty exciting for medicine. Now, these kinds of DNA tests are in fact available to consumers. If you've been watching the internet, you will notice that there are companies like the ones you see here, Pathway Genomics, 23andMe, DecodeMe, Navigenics, KnowMe, which will offer to you for varying prices as little as $99 in the holiday special that 23andMe was offering, to as much as thousands of dollars in some of the others. And these are done without going through your physician. This is direct-to-consumer marketing of DNA testing. And some have argued, well, let's get started with this and see what you can learn from it, because you can get useful information about future risks of illness, but it's still early days, and that information is going to be rather imperfect. So it needs very much to be a buyer beware. Some people feel this is premature. For me, I kind of figure if people are well informed and they want the information and it's possible to do so, who are we to be paternalistic and say, well, no, that's really not something that would be good for you. And this will give you risks predicted for things like diabetes or heart disease or Alzheimer's disease. Some of these are risks you can do something about. Some of them you can't. Do you want to know if you've got a risk for Alzheimer's disease? If there's nothing that can be done about it, people will vary a little bit in their responses. I'm curious, if we did a little poll right now, if it turned out that one of these companies was willing to give you all a free test as the result of being here this afternoon, would you sign up? Or is this information you want? How many people would want the information? Wow, a forest of people who really are interested. How many people definitely don't want the information? There are some people who are pretty sure they're not ready for this, including my wife. Whoa, what does that say? Well, she's a genetic counselor, so she knows a lot. Pay attention to her. Well, me, on the other hand, I decided to find out because I figured, you know, I'm curious, and I'm writing a book about personalized medicine, so how can I not sort of put myself through this? So I actually did send my DNA to 3 of these companies. I didn't tell them it was me because I figured they might notice that and do something a little different with that sample. And it was sort of gratifying to see that their lab work all came out the same. They were pretty good at getting the right answer in terms of did you have a T or a C in that position. Their interpretations, though, were not all the same. And I think that means that this is a field that's moving very quickly. And depending on which of the 25 papers published in the last 2 weeks you were using and which ones you weren't, you could get a higher or lower risk for a particular disease. So here's part of my report. This is the 23andMe report. This is the kind of thing you see on the website after you've actually clicked on it to see what your results were. So I think you can see there I've got an increased risk of atrial fibrillation. Just as soon not have that. Type 2 diabetes, that one really got my attention because that's a disease that really I would like not to get. And it actually happens to be the disease that my research lab at NIH primarily works on. In fact, the genes that they test for type 2 diabetes, some of them were discovered in my own group. And it's sort of weird to now look at this and go, gee, how did I turn out? So this one motivated me. And in fact, that's what I looked like when I did the test. And that's what I look like now. I'm a little grayer, but I'm also 25 pounds lighter. Because if you want to prevent diabetes, you should pay attention to diet and exercise. And I can tell you I was certainly not paying attention to either of those. And so this was a motivator. Now, is that a good enough reason to sort of lay off the honey buns and try to get some control of your diet and to begin, as I have done, on an exercise program with a trainer? No, I should have done that anyway. But for me anyway, this was the push I needed to recognize that, you know, immortality is probably not going to be part of my future. And if I want to try to stay healthy, I have some responsibilities to do so, and this pointed to some specific risks that I really wanted to avoid. If you want to read more about all of this, there's more about me and my family and lots of other things in this book, The Language of Life, that was mentioned in the introduction. Now I want to, though, quickly address something that people may have worried about is this information safe? Suppose you had that test, and suppose I gave you your results, but I also told your insurance company or your boss, would they then use that information against you to deny your health care or deny you a promotion? Well, I'm happy to say that that is not legal. It's not possible for that to be done without criminal consequences because of this bill, the Genetic Information Non-Discrimination Act, GINA. Some of us worked on this for more than a dozen years, and on this happy day of May 21st, 2008, President Bush signed into law this bill, which makes that kind of genetic discrimination illegal, at least for health insurance and for the workplace. It doesn't, however, cover life insurance. It doesn't cover long-term disability. So it is still something one needs to think about when you're having one of these tests. Is this the kind of information that you want to be sure doesn't fall into the wrong hands? Well, let's talk a little bit about how this all plays out in the media. And there's no place that's scarier in the media than this one. So take a little TV break here for a minute and listen to Stephen Colbert try to trip me up. Boy. There is no experience more white-knuckled than that one. Well, let's now try to draw this together in this wonderful way that JMU has been doing over the course of these months, because here I've been talking about science and then we got into the media. I don't know how we got there, but let's talk about the way in which science and the visual arts can come together, and I just have a bunch of images here of the way in which the double helix has found its way into lots of different perspectives, including body images like the tattoo on this young man. And everywhere you look these days, this seems to be all around us. Here are some architectural representations, including this. That is an actual street in Huntsville, Alabama, that is made to look like the double helix. It goes on for half a mile. And these other really quite interesting representations that you see here. But I guess JMU has taken this to a whole new level. And the way in which this remarkable collaboration between all of the orientation leaders and the Liz Lerman folks gave rise to this animated version of the helix, which is just unlike anything I have ever seen. So you all deserve a lot of credit for really, really making this into a human experience and having this be an appropriate kickoff of what is turning out to be an amazing year. But I want to show you one other example. Oh, wait a minute. There's, of course, what we're being experienced here also this afternoon and this evening in the performance, and also performances that have been going on yesterday and will go on again tomorrow, the Liz Lerman Dance Ensemble and this remarkable performance called Ferocious Beauty, which brings together a lot of the science I've been talking about with the artistry of their really remarkable troupe. But now look at this image. This is actually a piece of art that I am extremely fond of. And the artist who painted this is actually here in the auditorium. About 10 years ago, I got in the mail an envelope, inside of which was a 2x2 slide with a photograph of this painting. The painting is called Jacob's Ladder. Those of you who know something about the Old Testament will know about the story of Jacob and the dream and the ladder that he saw. Well, of course, in this case, the ladder is this ladder of the double helix of DNA. And this painting was put together in a very thoughtful, artistic, evocative way by Dr. Crystal Theodore, who's sitting right over here. I met Crystal Theodore for the first time today. Although we corresponded by mail and spoke by the phone, and my wife particularly got involved in speaking to her several times about this amazing piece of art. And she graciously, deciding that she didn't have another use for it, sent it to me and said, "It's yours." And so if you want to see where it is, well, my office on the NIH campus is decorated by that very same portrait. This is my desk. This is where I spend many hours every week. And when I'm sort of exhausted by the experience, I look up and I get enlivened again by Dr. Theodore's really wonderful Jacob's Ladder right there in front of me. And it certainly reminds me of the way in which the study of this particular science is all entwined in aspects of medicine, but also aspects of the humanities, of art, of our hopes for the future, of our way in which we're connected to family. All those things all wrapped together as this image can make you see. And this image appears in a few other places sometimes too. This you heard about, I have a guitar. Yes, and that is it. And you can see the double helix on the fretboard created for me by the Huss and Dalton luthiers down here in Stanton, Virginia. And in fact, as Linda described in the opening introduction, music has been very much an important part of my own experience growing up in the Valley. The chance to be in the Collins family with all of the amazing things that would happen in theater, in music, in the arts has greatly enriched my experience. And has also, because of that music, given me a chance to make a fool of myself in front of all kinds of people. So here are just a few photographs from that. Yeah, that is Chris Christopherson. Yeah, that is Joe Perry, the lead guitarist from Aerosmith, excuse me. And yes, that is the Edge, the guitarist for U2, in my office playing my guitar. Hudson Dalton was pretty excited to get a copy of that one. I don't think they quite expected the Edge to lay his hands on one of their instruments, but he did. So yeah, this has been also a wonderful kind of outcome of the glorious way that I had the chance to grow up here in the Shenandoah Valley. And so, now having titillated you with all that, it seems like maybe we should have a song. So what do you know? And yes, you can see this is the very same guitar with the very same double helix on the fretboard. And of course, every guitar has to have a name. B.B. King has Lucille, so I had to have some sort of a name for the guitar. And I actually had a contest amongst the people who worked on the Genome Project about what should we name this guitar, and lots of entries were provided. And it turned out that one particular suggestion appeared over and over again and made the most perfect sense. So, this guitar is Rosalind. And of course, there has a little As You Like It connection there, but actually it's a different Rosalind. So those of you who know the story of the double helix and its discovery by Watson and Crick will know that Watson and Crick were theoreticians. And in order to come up with the right answer, they had to look at somebody else's data. It was Rosalind Franklin who took that amazing X-ray picture that revealed the double helical structure that they figured out and put together their model. Rosalind Franklin sadly died of ovarian cancer only a few years later, before the Nobel Prize could be awarded. So the prize went to Watson, Crick, and Wilkins, and many of us wish that it had gone also to Franklin. And so this is Rosalind. So I'm going to sing a little song, and then we're going to wrap this up with a few questions if you still have the energy for that. But I need a little help on this song, because I've got a little bit of a cold, and at one point in here, there's a high C sharp. I mean a high C sharp. And I'm going to need a little help on that. And you will say, well, wait a minute. How are we supposed to help with a high C sharp? You'll know this when it comes. You'll just feel it. And I'm counting on you to do the right thing and join in and help me out so that if I screech really badly, it won't be quite as awful. So what is this song about? Well, it's a song about personalized medicine, of course. What else? So this is a song that you might imagine being written by somebody maybe 2 or 3 years from now who just got his complete genome sequence determined, decided it was time, and sent in his money, and you know, back comes the password saying, okay, it's ready, have a look. And so he's sitting in front of his computer looking at screen after screen of A's, C's, G's, and T's going, what the heck is that? And this might be the song he would sing. As I walk along the bases in all 3 billion places upon my computer screen, am I built for strong endurance or loss of life insurance? Am I a mere machine? I'm a walking through the genes, don't know what all this means. Oh, what can the meaning be behind that G and T? And I wonder, help me now, wonder why, help, why, you've got an A, I've got a C there. What does that say? Amazing DNA, DNA. So I'm glad to know I've got some of what Crick and Watson found and brought them fame. JMU, you found alliance between art and science. they're really much the same. I'm a walking through the genes, don't know what all this means. Oh, what can the meaning be behind that G and T? And I wonder, come on now, oh, wonder why? Why? You've got a C, I've got a G there. What does that say? Amazing DNA, a D-D-D-D DNA, a D-D-D-D DNA, a D-D-D-D DNA. Woo! Thank you all. Thank you very much. Oh, thank you so much. Thank you. Well, thank you. And if you're able, I gather we could take a couple of minutes for questions, and there's supposed to be people with roving microphones, maybe one down here and one up there. And if you have a question, raise your hand, and I'll try to point to you, and we'll get a microphone to you so we can all hear the question. It's a little bright up here, but here's a question down here. 1 second, the microphone is on its way.
I understand, brother.
This is a question about heredity, I can tell. Yes, Brother Brandon, your question now.
In the chromosomes, there is an end called telomerase. And I also understand that there is some research afoot that is looking toward trying to keep that from declining as we decline in age.
Yes, indeed.
And I wonder if you could comment briefly about that.
Not that any of us are interested in aging, since I, as the youngest brother, have now hit 60. We seem to talk about this a lot more than we used to. So yes, what is telomerase? Well, at the tips of the chromosomes, there is this repeated sequence that needs to be there to keep the edges from getting eroded. It's like, do you know what an aglet is? On your shoelace, it's that like thing right at the end, that little plastic that, or sometimes it's metal, that keeps the shoelace from getting all ratty. Well, your chromosomes have those too, but they're very special DNA sequences. And the problem is every time the chromosome gets copied, it gets a little shorter. Telomerase is the enzyme that fixes that, but guess what? Most of your cells don't have telomerase anymore. And so this is one of the major ways in which cells basically run down and can't keep dividing anymore because your telomeres get too short and then that's it. So there's a lot of interest now in trying to reactivate telomerase so that your cells could manage to keep going a lot longer and wouldn't basically run out of DNA. They don't run out of gas, they run out of DNA. And this is one of the most promising areas of research right now in aging. Do you all want to live to be 200 years old? Well, I heard a yes. As long as it's quality life, right? Well, that would be important. And it would not surprise me if in the course of the next many decades, we figure out how it might even be possible to extend the maximum human lifespan, which currently seems to be about 100, and add to that. We've figured out how to do that with animals. By working with telomerase and other things, you can get mice to live 20-30% longer than they normally would. If you live 20% longer than you normally would, you'd be pretty famous, I think. There aren't too many 130-year-olds walking around to talk about that. Other questions? Okay, Rick. I always know that when Seaford asks a question, I'm in real trouble, so...
I'm interested in the specificity of speciation, why you can, we're so close to bonobos, for instance, but we can't mate with them, but apparently we could with Neanderthals. What's the mechanism for that separation, And if there is some Neanderthal DNA in humans, has that any significance for human destiny, do you think? Because it's, apart from simply mating, it's it's different from mutation in terms of how your destiny works out.
So there's a lot in that question, as I knew there would be. So I don't know, I didn't bring this up, and if you've been watching the papers, you may have already seen it, but Neanderthals lived in Europe, but actually also in parts of Asia and in the Middle East, but died out about 30,000 years ago. And the assumption generally had been, until recently, that they were a separate branch that really went way off in a different direction from the branch that led to Homo sapiens, and that therefore there had not been any intermingling of DNA sequences. But very clever scientists working with Neanderthal bones from several different caves were actually able to extricate DNA and to sequence the Neanderthal genome, which is amazing. 5 years ago, nobody would have dreamed you could do that. You would have thought it would just be too degraded. And so we have that. And what we can now say is that for Europeans and Asians, about 4% of our DNA seems to have come from Neanderthals. And this looks as if, therefore, it was probably an intermingling that might have happened in the Middle East, because that's the way in which these migrations occurred. If you're going to end up with those sequences in Europe and Asia, probably it traveled through the Middle East. Interesting, if you look at Africans, you don't find Neanderthal DNA. So it was clearly something that happened after the out-of-Africa migration. And that has caused a lot of people to think philosophically about what that means about our inheritance. Now, mind you, the Neanderthal genome and the Homo sapiens genome are extremely similar, because they really only separated less than 1 million years ago. And so you have to look very carefully to perceive that there is that difference there. But it is there. And does it say anything particularly meaningful about human biology? Does it say anything about Asians and Europeans? It makes for some really good jokes about, yeah, I knew you were a Neanderthal all along. And you can use that one on somebody as long as they're not from Africa, in which case you're wrong. And the other part of Rick's question was speciation. So how is it that you get to the point where species can no longer keep mingling like this? Clearly, Neanderthals and humans were able to do so because they're very close. Basically, it gets to the point where if the chromosome rearrangements are sufficiently different, actually in the process of going through generations, chromosomes have to pair up together. Chimps and bonobos actually have one more chromosome than we do, because our chromosome number 2 is broken apart into 2 chromosomes, or rather theirs were the ancestral ones and ours is one that has fused together. And that means things don't match properly when you're going through something called meiosis. So speciation happens when the deviation between 2 species gets far enough apart that they can no longer match up at the time of making sperm and egg and produce something that is going to give rise to a stable outcome. And speciation is fascinating because it takes more than just time for species to develop. It takes some kind of geographic separation, because otherwise you continually get crossbreeding and you never have enough differences built up so that species can happen. You've got to have both time and a geographic separation, like a mountain range or an ocean. And then this whole process happens. And we're learning a lot. I mean, that Darwin, I think, amazingly, thoughtfully predicted, but now we're really beginning to grasp those details. There's another question back here. And I know we shouldn't go on too much longer because there are other things that are supposed to be happening, so I'll take one more question after this, including if there's one up in the balcony. I do see one up there, so that'll be next, and then we'll shut her down. Yes?
Saw a video recently where Christopher Hitchens mentioned you, and UVA hosted Richard Dawkins last year. I'm wondering if you can comment at all on the debate with regard to science and religion, especially as polar as that seems to be, with perhaps Dawkins on one side, you and people like Ken Miller on the other.
I debated Richard Dawkins a few years ago, and you can still go to the Time magazine website and look up that debate, because they published it, quite a few pages of it, as a cover story in Time. And I think it's a good representation of the different perspectives that he has and that I have. Certainly there are strong voices now that are arguing that science is advanced to the point where it is no longer necessary to consider the possibility of God, And certainly Dawkins argues that strongly. Christopher Hitchens, in his book, God is Not Great, argues it as well. Both of them also argue that religion is not only not necessary, but that actually it's the cause of a lot of the harm and evil in the world. I have a different view. I was an atheist for a while. I became a believer in my 20s. For me, I see no conflict between science and faith. And I see that science is basically unable to answer some of the questions that I think are most interesting. And I don't want to miss out on those. Science doesn't help with the question about why am I here, or what was there before there was something? Why is there something instead of nothing? Is there a God? What happens after you die? Those are all questions that science basically has to remain silent about, because they're not about nature or the natural world, and science's province is the natural world. I think it is entirely possible to be both a rigorous scientist who will not accept your conclusion unless I see your data, if it's about questions about nature, but also to believe in a creator God who started this whole enterprise with a purpose and who worked through the process of evolution to create this amazing creature called human beings, and intended that to be a relationship that was involving the possibility of connecting human beings with God the Creator. And I know of no compelling argument to say that that's not entirely plausible. But it is, of course, a matter of faith. It's not something I can prove, nor can anyone else. Christopher Hitchens has become a close friend, even though we are widely different in our views here. Christopher is, at the moment, suffering from a fairly advanced case of esophageal cancer and is in considerable difficulties as a result. And I've been trying to help him, as he lives in Washington, to negotiate through a whole bunch of options that he might have in terms of the management of his disease. And it has been a great experience to get to know him better as a prodigious intellect, but who happens to come down in a very different place about this question. I actually think that one of the great tragedies of the current time is that this perspective about science and faith has been so dominated by the extreme voices. There are those arguing on the one hand that religion is unnecessary and evil and irrational, and saying so sometimes quite shrilly. On the other hand, there are people arguing that fundamentalism in religion is the only really acceptable form, and if you are one who actually looks at the scientific evidence for evolution and for the age of the Earth and concludes that there's very compelling evidence there that the Earth is actually about 4.5 billion years old, that you're not a true believer, and that therefore you are at risk of a bad outcome in the afterlife. It does seem to me that both of those extremes are unnecessary, and most people are uncomfortable with them, but there hasn't been a whole lot of discussion about how to harmoniously blend the scientific and the spiritual worldview. For me, that blending is enormously satisfying. And that's the point of view I've tried to carry across, although it's occasionally gotten me in hot water with the people who are coming at this from the other perspectives at both extremes. So there's a question up here, the final question, somewhere. Yes?
There are a number of cases where there have been acts of generosity across species lines, where people who do not have genetic relations or close ones, or even mimetic ones in terms of sharing of ideas, will recognize qualities of spirit or some nobility in the actions of another sentient being, and put themselves at risk at times to come to the aid. So my question is, do you see the possibility that much as minds create memes and bodies create genes, that there may be a triad in terms of spirit creating seeds as well that we're prepared to help see flourish?
Wow, that's a fundamentally profound question about altruism, really. And this is a very active area of current discussion between science and philosophy and theology. If I'm walking down the riverbank and I see somebody out in the middle of the river who is clearly in trouble, calling for help, who I don't know, I feel this urge to try to help, even if I'm a poor swimmer, and even if the current is rapid and I know that I might also perish. I feel this calling to do something, to jump in and try to save that person. That's an interesting thing to describe, because evolution, pure and simple, would probably not approve of your putting your DNA at risk in order to try to save that other person who you don't even know. Evolution would say, hey, survival of the fittest. That person is apparently not living up to that expectation. Let him go. But we don't feel that way, do we? Amazing story of a construction worker in New York standing on the subway platform as the train was approaching, and a young man next to him who he'd never met. He was black. The student was a white male, went into a seizure and fell onto the tracks in front of the oncoming train. A construction worker standing there with his 2 young daughters quickly handed them to the next person and jumped onto the track. It was too late to be able to pull the young man out, so he basically pulled him in between the 2 tracks, covered him with his own body as the train rolled over them. Miraculously, there was just enough clearance that they both survived. Now, what was that about? What would cause somebody to take that kind of risk for someone they didn't even know, and certainly someone of a different cultural group, as we subsequently learned when these families got to know each other and discovered how different they were. And yet, we admire that, don't we? You read about that and you think, well, that's the human spirit in its noblest form. I think that's interesting, because that is not an action that seems to make much sense from a pure evolutionary perspective, where passing on your DNA is supposed to be the most important driver. If you were looking for a glimmer of something that might point you towards a force that is noble and holy, and you find that same glimmer in yourself in this kind of exceptional circumstance, seems like that is worth paying attention to as a signpost of some sort. I'm not going to say that proves anything, but I think that is a dilemma for those who are completely sold on the idea that evolutionary naturalism can explain every aspect of humanity. There's still some mysteries there, and isn't that a good thing? And isn't it a good thing that those mysteries can inspire both science and art, and the kind of art that has been existing for all these centuries and which is now being demonstrated here at JMU during this wonderful celebration? Also reminds me that there are aspects of humanity that make no scientific sense. Why do we care about beauty? Why are we so attached to the idea of seeking it out? It's not clear. Why does music inspire us? What is that about? It's very difficult to come up with purely naturalistic explanations. And yet this seems to touch something deep within us that is calling us to some kind of other understanding than one that is solely based upon molecules. So I would encourage you during the course of this thoughtful day and evening to think about all those things and the way in which science and art can teach us, but maybe can also ennoble us. Thank you all very much. Thank you.